Hi, my name is Eva Prašnikar. I'm a young researcher at the National Institute of Chemistry in Slovenia, and today I'm going to talk about molecular simulations of the HLA β2m peptide NKG2A CD94 immune complexes. We will start with a short introduction involving senescence, senescent cell accumulation, and immune complexes. Here you also highlight our main objective and proceeded with methods, results, and conclusions. Aging represents a greater susceptibility to age-related diseases. Senescent cells are one of the general hallmarks of aging. These cells are permanently arrested in G1 or G2 stage of the cell cycle, accumulate with age, and supposedly contribute to the development of age-related diseases. The most partial reason for the senescent cell accumulation is the formation of the immune complex between HLA ligand expressed on the senescent cells and the NK cell inhibitory receptor NKG2A CD94, which inhibits the NK cell cytolytic activity. Here, recognition and inhibition are dependent on the non-numeric peptide incorporated into the ligand counterpart between alpha-1 and 2 domains of the HLA heavy chain. These peptides are usually derived from the leader sequences of other HLA molecules. There are several peptides that enable immune complex formation and sequential inhibition of NK cells, for example peptides G and B7, but there are also peptides that do not enable the inhibition of NK cells, such as the HSP60SP peptide and the mutated version of the B7 peptide. So our main goal here was to uncover the mechanistic details of recognition between receptor and ligand counterparts leading to NK cell inhibition. So we built the immune complexes based on X-ray structure with resolved extracellular domains of receptor and ligand proteins. We built 4 immune complexes differing in the identity of the non-numeric peptide, whereby according to the experimental data, 2 of them enable inhibition of NK cells and 2 of them fail. We then ran molecular dynamic simulation and obtained trajectories with 1 microsecond in their equilibrated production time. We then visualized trajectories, calculated cross-correlation matrices and free binding energies, and compared the obtained results for each immune complex. No major conformational changes were observed with cross-correlation matrices. We observed similar correlation patterns in models conferring NK cell protection and in models with absent NK cell protection. Nevertheless, there are more pronounced patterns present in the models conferring NK cell protection. From most representative clusters, we notice the positional deviation in the amino acids at the 5th position of the peptide. In the models lacking NK cell protection, the distance measures between C-alpha atom and HLE were greater compared to the models conferring NK cell protection. We also noticed the increased flexibility in the HLE alpha-2 domain near the C-terminal site of the non-numeric peptide. In the models conferring NK cell protection, a specific hydrogen bonding network was found. Namely, the peptide forms interactions with ligand and receptor proteins, and a positional deviation of arginine of NKG2A protein was noticed, and then the specific formation of the H bonds between both receptor proteins was found. So we hypothesized the following sequence of the events. Contact between CD94 and backbone atom of the peptide positions the CD94 residues. And then the peptide H bonds with residues of the HLE alpha-2 domain drives the placement of the alpha-2 domain. The alpha-2 domain then influences arginine 147 of the NKG2A protein in its vicinity, which subsequently contacts lysine 145, and so formation of the key interaction between NKG2A and CD94 proteins is enabled. Then pairwise decomposition of the free binding energy reveals key interaction for receptor-ligand binding, which are presented in the table on the right. The pair residue decomposition of the free binding energy revealed key residues for receptor-ligand binding, which are also presented on the table on the right. And further on, we noticed that the CD94 forms the majority of the energetically important interactions with the ligand. So in summary, our study has shown that key interactions between receptor-ligand complexes are fine-tuned by the ligand-specific monomeric peptide through an entangled network of hydrogen bonds. Then NKG2A regulates NK selectivity via key interactions with CD94. The CD94 receptor and HLE-alpha-1 domain form the bulk of the energetically important interactions that drive the formation of the immune complexes. Further on, our simulations could serve as the basic for future experiments and for rationale-based drug discovery for external modulation of NK cell activity.